Assalamu alaikum. This is the second portion of our lecture 1. Before going to the basic terms of biotechnology and genetic engineering and the recent discoveries or the ancient discoveries and roles of several scientists, I want to show you a specific picture which is one of the most familiar picture for all of you. From school level, you are learning how to draw a cell, whether it is from animal source or plant cells or plant source. And of course, you have the idea of basic differences between animal cells and plant cells. As we are learning about genetic manipulation of cell or cellular organisms, so we must know or we must recall the name of the organelles and their particular functions of an animal cell. So let me remind you again, this is an animal cell. An animal cell does not contain any cell wall, which is a basic feature of plant cell. Another difference between animal cell and plant cell is that they have a larger vacuole. You can see this organelle. This organelle is very smaller here in animal cell, but it contains the largest portion of a cell, like more than 50% or up to 50% portion of a cell, so that the nucleus of a plant cell is slightly moved to the edge of the cell. However, as we are learning about animal cells or we are seeing the functions of animal cells, let me read out the whole organelles. Mitochondria, another common thing, which is the powerhouse of a cell. Also, it is important for carry out certain important reactions of our biological metabolism, such as oxidative phosphorylation and electron transport chain. And few of the reactions are known to you, and the rest we will learn gradually. Centrioles. This organelle give the structural strength of a cell. Ribosomes, the one and only organelle that is present both in eukaryotic cell and in prokaryotic cell. Chromosome, that contain our genetic material or the DNA is compacted within the chromosome. Endoplasmic reticulum, nucleus, nuclear membrane that protect or that create a channel between cytoplasm and nucleoplasm. Golgi apparatus, the protein sorting organelle. Vacuole and cytoplasm, that is the liquid portion of the cell carrying the whole organelles. So now you have had the reminiscence of the function of animal cell. Now let's proceed forward. Here another term has been added to our lecture, which is genetic engineering. Till now we are having the brief idea about what is biotechnology, how it is advanced from ancient period to modern era, and we have also learned the basic or the key ingredients that transfer or that switch biotechnology from ancient or classic period to modern period is genetic engineering. Here the same term or same thing you will see again. So the examples are the application of biotechnology is bread making or wine brewing, cheese and yogurt production, or classical plant and animal breeding. But recently biotechnology has been diversified to numerous applications. The application of biotechnology is not or is no more confined within this. That means from bread making to animal breeding. Now biotechnology has rendered their branches in every sector of life, from pharmaceutical to medical applications to forensic and also to gene cloning. That is written here. Biotechnology has recently developed and diversified to numerous applications, and the key to this development is the advance in genetic engineering. So we learned that genetic engineering is the key in advancement of biotechnology. Now, what is the basis of genetic engineering? There should be some particular theme or particular basics that help us to find the way of engineering the genetic constructions or constituents. What is it? It is the recombinant DNA technology or recombinant DNA techniques, RDT. This is the most important phrase or term, whatever it is, please keep in mind that through the help of recombinant DNA techniques, we learned we are able to engineer a genetic construction of any organism or plants or even animals. So this is the blessing in case of biotechnology. So what is the chain? The chain is The chain is like that. Through recombinant DNA technology, we know how to genetically engineer a cell, and through the help of genetic engineering, we have some advanced or some recent new applications of biotechnology. So it's a consequence from RDT or recombinant DNA techniques to genetic engineering, and through genetic engineering to modern biotechnology. Please keep in mind or know what is this flow. Recombinant DNA techniques are important tools for conducting genetic engineering. So what are the important tools for conducting genetic engineering? Of course, the answer should be recombinant DNA techniques. Please do not be confused here in this slide. Sometimes I ask you the question that what is the key ingredient or the key terminology for biotechnology? The answer should and must be genetic engineering, not recombinant DNA technology. If I ask you the question, what is the key for genetic engineering, then you have to answer it is recombinant DNA technology or recombinant DNA techniques. There is a staircase or a step. Step 1 RDT, step 2 genetic engineering, and the step 3 is modern biotech. And I think by this time it is clear to all of you. Let's have a look at the scientists or the renowned personnel through which or by the hard work of which personnel now we are having a highly modernized or highly scientific lifestyle. Louis Pasteur, biotechnology and their historical milestones. Who is Louis Pasteur? A French scientist. And he asserted that microbes are responsible for fermentation. And I think all of you have at least a bit of idea about what is fermentation. Well, let me explain it again in a short form. Fermentation is a procedure of converting a raw material or an organic substance into a better version or a simple version of that particular substrate or a newer compound with the help of microorganism. That means microorganisms will carry out certain reaction on a substrate through which they will get energy or food source for their development, and at the same time they will produce some byproducts that are not useful for them, and these products will be utilized by us, or these products will be our desired products or biotech products. This is the basic theme of fermentation, and he asserted first that it is the microbes that are responsible for fermentation. That means in ancient period fermentation was there. They produced beer from rotten vegetables or rotten fruits, and they know that some kind of reaction is being occurring here, and that is fermentation reaction. These are the products of fermentation, but they have the least idea about that microorganism, or there are some microbes present there who is carrying out the whole reaction to give us such brewery or other food compounds. So it is Louis Pasteur who first asserted that microbes are responsible for fermentation. Now here in these points, let me make you clear that from any slide of my course BTE 101, I will tell you to learn or to know the discoveries of scientists, but their time period or the dates are not so important. You just have to know that yes, Louis Pasteur did asserted the responsible agents are microorganisms for fermentation procedure. You just have to know this discovery, but in which period or which time it is not mandatory to memorize, because it is not possible for a person to memorize a huge number of dates or a huge number of numerical values. However, you must know the discoveries of particular scientist. For example, if I ask you that who first invented pasteurization, not even pasteurization, who first claim the importance of microbes in case of fermentation, if your answer is Karl Ehricke or Robert Koch, then that won't be justified, right? You should know that it is Louis Pasteur that or who first find out the responsible agents against fermentation reaction, which is the most important reaction in case of biotechnology, both in industrial aspects and in biological science aspect. However, in 1863, he invented the process of pasteurization. Now, what is pasteurization? In our house, when we buy some milk, liquid milk from groceries, we frequently or often we see the terminology used, pasteurized milk. Now, what is this pasteurized means? Pasteurization is a procedure of killing or eliminating the harmful microorganism from milk that may cause cross reaction or that may produce some toxic compounds within the milk. So through either heating or applying other procedures, these microorganisms were eliminated from the milk before they are being marketized or commercialized. So he invented the process of pasteurization, heating wine sufficiently to inactivate microbes that would otherwise turn the wine sour. That means there were some microbes present in wine that may have turned this wine sour or may have the capability to destroy the texture or the taste of wine. So he heated this wine in a sufficient temperature in order to kill that certain microbes, but at the same time he maintained the appropriate condition that so that the flavor of the wine remains same. That is why pasteurization has only effect on destroying the harmful microbes but not removing or reducing the texture, flavor, or other good criteria of that particular food. So this is the invention or the role of Louis Pasteur. From the next portion, we'll know about certain other scientists and their discoveries.